All right. Well, hello, everybody. This is the walkthrough video for Wednesday. It is March 26th in Try 3 Week 5. And we're finally in our heredity unit. So I get to show you these goats that someone drew on the whiteboard once. They're just a little bit different. Aw. Hey, here is a question that is a super leading question, I think. So go ahead and write something about it so that if you see this thing, you will remember it on a test. Cheers. Today we're going to think about how we can, it is neither of those things are true. Today is my big kid's 11th birthday. That is amazing. Happy birthday, Nomura. Today we're going to predict the outcomes of genetic crosses. We're going to review Mendelian genetics, which is actually still used lightly often in science and medicine, though simple ideas about a trait being dominant or recessive are actually not nearly so interesting as the other rich factors and mechanisms that can make diversity in living things. So, as I promised you, we are going to press pause on all the project stuff because I do think I looked and I think you do want an in-person facilitator for those things. So, what we're going to do is highlight some of the work from Unit 5 about heredity that kind of stands in and thinks like allows us to connect what we've been learning in Project 3. So today's assignment is pedigree assignment. It looks like a worksheet, but you are just going to answer the questions in your lab notebook this time. As promised, all work this week is either digital or done in your lab notebook. So here is our Unit 5 guiding question. And one of the things that we talked a lot about is traits, right? And like those physical characteristics. But it turns out that not all inherited characteristics get expressed in the next generation. You can get all this diversity. Do you remember the pigeons that the bio students did that are in my room, right? From identical pigeon parents, you get this exceptional diversity of pigeon outcomes. So, will you please pause the video and do what it tells you to do here, including the table of contents? Okay, great. So we're going to think about pedigrees, but I wanted to help you remember some ideas about Punnett squares. So if you have ever looked at people with eyeballs, you know that there are lots of different colors of eyeballs. There are not just blue eyes and brown eyes. The last time I checked, scientists thought there was something like, is it like 12 or 14 different pigments? Whitney's nodding. We're in our hotel room doing work, homework. It's 9:30 on Tuesday. What's up, children? But we're going to pretend in this Punnett square that there is one allele for brown eyes, one allele for blue eyes, and that these are your choices, right? And I wanted you to remember that heterozygous parents or ancestors or the where the gametes come from that makes the next generation make this probability map where there's a 1 in 4 chance of an organism in the next generation having blue eyes in this case, right? Now, this isn't a checklist. It's if there are 4 kids, it's not guaranteed to be a 3 to 1. In fact, they might all have light colored eyes. They might all have dark colored eyes in that next generation of like peccaries or polar bears or whatever. But this idea about where they come from, the reason that the square looks like a square and that all of these are square is because we're pretty much telling you the genotype of the adult somatic cells, the body cells. Each of these areas is split 50-50 this way because we know that there is a 50-50 chance that you're going to get either one of the gametes. The gamete that you get in the next generation is a 50 chance you'll have either of those alleles. And that's different from like if you looked at the gene pool, like we talked about in Unit 7, like let's say that an allele is really rare, it's dominant, but it's over here to show you just how rare it is. Depending on how rare this allele is, the chances of you being heterozygous and homozygous dominant, either of those genotypes, and having a dominant phenotype is pretty small. And even small changes here change the probability that you would be homozygous for whatever that uncommon allele is. So you can't just like average when you think about big populations. The only reason we can make a Punnett square that looks like it's two up, whoops, which slides two up like in that grid and that square grid is because of that. So, all right, these are karyotypes. Remember we learned about karyotypes. You don't need to write this down, but pedigrees, what we're going to learn about and review today, they trace an inherited genetic condition from ancestors to descendants. They determine if a condition is inherited, if a condition is dominant or recessive, and they use standardized symbols. And I want to help you remember that we know a condition is dominant if it's got either one of these genotypes, right? So like big A blank is a dominant genotype because it gives rise to a dominant phenotype, a dominant trait, a dominant physical characteristic. Pedigrees use standardized symbols, and so it's important for me to pause for a moment once I get the slides to be the right size. There we go. That families come in all shapes and sizes. And so I wanted to make sure you knew that a pedigree chart is not a family tree. It only represents the passing down of genetic traits from ancestors to descendants. Um, the people that you live with and call family could be very different from house to house. People are adopted, which is wonderful. People use reproductive technology, and that's something that's increasingly common now. So, this idea about whether or not it's a family tree, they do not represent that. They also only show phenotype and not genotype unless otherwise indicated. So, like most human and non-human individuals never even get a karyotype. And so you're looking at sex assignment at birth rather than chromosomal sex, which it turns out can be different. And so this type of chart is what they look like. And what I'd like you to do, I gave you the link for this. It's set so everybody can see it. I'm going to ask you to copy those into your notebook. You probably want to use a feature that looks a little bit like this. So go ahead and pause and make this list. It should say pedigree key in your lab notebook. And then go ahead and copy the key to the symbols here. Cool. Now, it turns out this is a four-generation pedigree. And so, in my family, my kids have cousins because of the way that our family is put together. They have cousins who are graduating from high school right now. So they're like big age differences. So like we're never going to show you those. We're only going to show you ones where the generations are like very, very clear, right? And as you can see, these are kids that come from these two, you could call them genetic parents or ancestors. We draw a line to show that the gametes have mixed, and we draw this straight line and then a bunch of branches off to indicate those siblings together. These are siblings. Please do not draw them holding hands because then it would mean that the gametes have mixed. And in nearly any pedigree that you are going to see in any circumstance, that's not what happened. We have a lot of different ways that scientists have represented the beauty and complexity of ancestry, identity, and family life. Lots of different things. People of unknown sex. I don't know if you know, but there are, especially when people are receiving genetic counseling, these are particularly important to track, and we have special symbols for those. Just lots of interesting things to like think about that diversity. This is something that was published in 2022 in the National Science Teachers Association professional journal. This idea that you begin to have students use some more of these symbols. This isn't perfect for the reason given here, but it's like still a pretty good starting place. One more time, families and reproductive technologies come in all shapes and sizes. Okay, so I'm going to show you how to guess and check. Basically, starting here, you're going to follow the instructions. So right now, you're going to write this down, and you can use this one or the information up here, either one, to draw that pedigree. Hopefully that was straightforward. So pause the video and do that now. All right. Next up, oh, I forgot to tell you the slides are here, so you can access the pedigree slides that I was just showing you. Sorry, pals. All right, so next up, dominant or recessive. You're going to assign genotypes below each person in little writing. So you're going to draw the pedigree out, and then you're going to write a couple words about whether it could be dominant or recessive. If you don't need to draw it out twice, you don't have to, but quite a few people do. So it's up to you what you would like to do about it. So if the condition is dominant in this case, you know that the filled in circle is big A blank. You don't know whether that person is little a or big A little a heterozygous or big A homozygous dominant. But like if there are no mutations, then you probably do, right? Because there's no place, if this condition is dominant and we assume there's no mutations from generation to generation, there's no place to get that dominant allele. One of these shapes would need to be dominant to make that happen. But if the condition is recessive, you could imagine that these two people, if it's people, are heterozygous and they each have a recessive allele to pass on here. If this is complex, you can go and watch, I think this content starts in 5.3, and you can watch the 5.3 video on AP Classroom. It's going to talk about dominant recessive pretty early on there. You're going to do the same for case 2. So copy this pedigree and then describe whether dominant is possible or recessive is possible. Heads up, if we assume no mutations, only one of those is possible. But for case three, without more information, you don't know. So I'm going to ask you, is dominant possible? Explain why. Is recessive possible? Explain why. So go ahead and pause the video. You're going to have at least one copy of each of these pedigrees plus some writing. Please do that now. All right, next up, this is the genesis problem. You're going to explain to a couple the probability of a child developing an autosomal genetic condition if their gametes are both used. Because little is known about the genetics of this condition, you're also going to characterize it to aid other medical personnel and geneticists. And just so you know, Kirsten Hall, Miss Hall's kid, is a sophomore. Maybe you know Cortland if you did marching band or Girl Scouts or the other things that Cortland did. She's studying to be a genetic counselor. So it's that's a major at Purdue. So hey, this is the information about spouse one and spouse two. And instead, we used one and two so that when you make the pedigree, you can label them as S1. Maybe this person is S1, maybe this person is S2 or whatever. So just so you are able to do that. And you'll see you've got some patient information here. Um, in the space below, which means on your in your lab notebook, you're going to translate the patient history, but you should not do it in your lab notebook to start. You should do it on a whiteboard or some scrap paper. So maybe you have a whiteboard in the classroom, maybe you're using some scrap paper, both of those are fine, but you are going to want to like proceed carefully. Once you make it, and especially if you talk to some other groups during classroom time, you're going to know if the condition is dominant or recessive. And you're going to, in your lab notebook, you're going to say this condition is recessive or this condition is dominant, and then describe why. Now, the last thing you're going to do after you add genotypes to each person in the patient history who's represented in your pedigree, so all the symbols are going to have genotypes. You might have some underlines because you don't know whether the second allele was dominant or recessive. And then you're going to draw some Punnett squares to answer question 4 and question 5. You do not need to use the explanation tool to write out a final answer. Once you're done with these, you can write end, and that'll end your work for today. All right, last but not least, why are pedigrees important? What do they let us do? 1 to 2 complete sentences, please. Hope you're doing okay out there. We'll see you soon. We won't. I'm still in Philadelphia, but you're great. Bye.